The last class, I have described about the overview of amino acid glycine. In today's class, we will discuss regarding the synthesis of glycine and also catabolism of glycine. These glycine is an important amino acid and most asked question in the final exams. And it is quite easy when we see when we look at this. First one, the basic thing you should know about a glycine is the simplest amino acid. First amino acid and simplest amino acid, and it is a non-essential amino acid. But what is this non-essential amino acid? So there are two kinds of amino acids. You see a classification, there are various types of classification. One of the classification where according to the nutritional value, either it is essential or non-essential. So it is a non-essential amino acid. It means it, it doesn't require to take from our diet. So it can be synthesized from our body. And next, it is a glucogenic amino acid. What is this glucogenic amino acid? So glucogenic, it means that something where the glucose has been synthesized, where the glycine is one of the amino acid where it involves in the formation of a glucose through a process called as gluconeogenesis. Now we will discuss regarding how the glycine is being synthesized. I already discussed, right? It is a non-essential amino acid. It means it can be synthesized in my body. So it can be synthesized from serine, synthesized from threonine, it can be synthesized from choline, and it will also be synthesized from a glycosylate. Okay, and we will see one by one on how the glycine has been synthesized. Now you can see the figure, right? If you see this flow chart, right, it's complete flow chart, you will understand the complete synthesis of glycine. You can write, even if you write the same flow chart in an exam, you will get out of out marks. We'll see one by one. It will be very easy to concentrate. You can see here, right? This is the basic structure of a glycine. This is the basic structure of a glycine. If you see the structure of a glycine, we have one carbon dioxide group, one mean not carbon dioxide, it is a COH group and one CH group, one ammonia, right? So it means that we can also synthesize the raw material from the raw materials. I mean, if we give a raw material, the glycine can be synthesized. So that is how you can see here the carbon dioxide and ammonia both combine in the presence of enzyme glycine synthase. What is enzyme glycine synthase? It means that we are synthesizing the glycine. And one more is missing. What is that one missing thing? One is the CH group. So who will give the CH group? So there are one carbon donors. So what is the function of one carbon donors? It will donate the one carbon group. So one of the one carbon donor is N5N10 methylene tetrahydroxyfolic acid. So it will donate one carbon group to the carbon CO2 and NH2 group and form as a glycine. So where N5N10 within tetrahydroxyfolic acid, after donating a carbon group, it forms as a tetrahydroxyfolic acid. This is nothing but a vitamin B9. Folic acid. The active form of vitamin B9 is nothing but tetrahydroxyfolic acid. And along with that, we require a coenzyme PLP. And the NADH is also a coenzyme where NADH is being converted to NAD because the hydrogen group is also donated by the NADH and taken up to form as a glycine. So that is one of the reactions where we give carbon dioxide and ammonia, and along with that, one carbon donor in the presence of enzyme glycine synthase and forms as a glycine. And another one is the threonine. So threonine by the presence of enzyme threonine aldolase. Aldolase is something like cutting. It broke down and form as a glycine. The next reaction is can also synthesize from a serine. You can see here it can also be synthesized from a serine. So you can see the serine. You see the structural difference between a serine and glycine. You can see here, right? Let me make it clear. See the structure of a serine and a glycine. There is an extra CH2OH group for a glycine. You can see the structure here, extra CH2OH group. So this if I remove the CH2OH group, now the rest of the thing is nothing but a glycine. So what I have to do? I have to remove. So So if I am removing this, someone should take, right, says to which, so it is taken up by again the one carbon acceptor. Here, no, tetrahydroxyfolic acid is acting as an acceptor and form as N5N10 methylene tetrahydroxyfolic acid. You can see here. Here. And what is an enzyme? Serine hydroxymethyl transferase. The name of the enzyme suggests that, see, there is a serine and there is an hydroxymethyl group has been transferred to what tetrahydroxyfolic acid to form as a glycine. And one more thing is like glycoxylate. You can see here, right? You can see it's a glycoxylate. So glycoxylate in the presence of enzyme glycine transamination reaction. Mean in the transamination reaction where amine group is donated to the glycoxylate and form as a glycine. Again, this glycine can convert back to the glycoxyl by enzyme glycine oxidase. And I already told you that it is a glucogenic amino acid. Why it is called glucogenic amino acid? You can see the glycine can also be glycine also synthesized. Serine also can be synthesized from a glycine. So, this serine can form as pyruvate by serine dehydratase, and this pyruvate can form as a glucose through a process called as gluconeogenesis. Process called as gluconeogenesis. And what is this ethanolamine? So there is a cycle called as serine, glycine, and choline cycle. Now there is a serine, glycine, and choline cycle where you can see the serine is converted to ethanolamine, and these ethanolamine can convert to the choline. Okay. So what is this choline? Choline is nothing but trimethyl ethanolamine. So if they add three methyl groups to the ethanolamine, it forms as a choline. And this choline is also a donor, one carbon donor, where it can donate the three methyl groups. How many methyl groups? Three methyl groups. Ultimately, there is that is a different cycle where serine, choline, and uh, glycine can form as a cycle. And these dynamics can also be converted to glycoxylate, and glycoxylate, uh, where this glycoxyl, you can see the glycoxylate is can either it can when it is broken down, it is formed as formate and oxalate. Formate and so it formed as formate and oxalate. This formate is also one carbon. It is taken up by the tetrahydroxyfolic acid and formed as N10 formate tetrahydroxyfolic acid, and oxalate is excreted in the urine. So this is all about the synthesis of a glycine. So glycine, how it is synthesized, can synthesized from serine, threonine, choline, and glycoxide, also from it is carbon dioxide and ammonia. Next, we'll discuss about the degradation. It means that how the glycine is degraded in our body. So you can see here, right, the glycine. So glycine, the branch of enzyme, glycine decarboxylase. So what it is decarboxylase? Is removal of carbon dioxide. So it requires coenzyme redoxyl phosphate. And if you see the structure of a glycine here again, you can see the structure, right? So if the carbon dioxide has been removed, we can see this carbon dioxide has been removed. What is left, one is CH group and ammonia, right? So that is what ammonia methyl group. So here is it is an ammonia and it is a methyl group. Okay. So ammonia methyl group is formed. This ammonia methyl group, okay, is been where it has been split into two compounds. One is ammonia, one is methyl group. So ammonia, you can see the ammonia, it is excreted in the urine. Okay, as in the urine, where it will go to the kidney and and excreted in the urine as a urea. We will study this in the urea cycle. Okay. Ammonia is excreted from a body as urea, and this one and one more thing is left. What is this methyl group? So who will take this methyl group? Methyl group acceptors. Yes or no? There is a methyl group acceptors. Right? Who will accept the tetrahydroxyfolic acid is accepting the methyl group and form as methylene tetrahydroxyfolic acid. And it is done by enzyme methylene tetrahydroxyfolate synthase. It is synthesizing methylene tetrahydroxyfolic acid. That's why the enzyme methylene tetrahydroxyfolic synthase. And there is one more hydrogen group, right? This hydrogen group is taken up by the lipoamide. Who will take the lipoamide and form as reduced lipoamide. This reduced lipoamide combined with the NAD, okay, where it will
transfer the hydrogen group to the NAD and again form as lipoamide, and this NAD form as NADH plus H plus. So this NAD, where what is the function of this NAD? Enter into the electron transport chain where it will produce 2.5 ATPs. This is all about the synthesis and breakdown of um, amino acid glycine. Thank you.